Hello, everybody. My name is Roland Schafleitner. I'm working at the World Vegetable Center in Taiwan as flagship program leader for vegetable diversity and improvement. Today, I have the pleasure to introduce you briefly to our Amaranthus core collections. Amaranth. Amaranth was originally domesticated in Central America, but is now considered to be a traditional African vegetable. We like Amaranth because it is tolerant to drought and to heat. This is mostly due to the fact that it's performing C4 photosynthesis like maize and other tropical crops. It is susceptible to only a few diseases and pests, means in practice we observe a very little pest and disease incidence. It is very versatile. We can grow it as a vegetable, or as a grain crop, or as we will see in a minute, as both. And most importantly, it is highly nutritious. It is rich in proteins. The grains have a balanced amino acid content. That means it contains all the amino acids that are essential for human nutrition. It is rich in minerals like calcium, magnesium, potassium, phosphorus, and iron. And of course, there are also many vitamins abundant in Amaranth vegetable and in Amaranth grain. Amaranth can be grown as a vegetable or as a grain, or Amaranth can be used for both. Grain Amaranth that is, can be defoliated and the leaves can be eaten. And in the chart below, it is shown that there are cultivars available where up to 50% defoliation does not lead to any significant reduction of grain yield. The current production constraints of Amaranth are a little bit pests and diseases, but this only applies if Amaranth is grown to maturity. On Amaranth plants that are grown just for 2 or 3 weeks for uprooting and as a vegetable, pests and diseases are rarely seen. There is still some work to do to increase the balance between grain and vegetable yield for dual use amaranths. Lodging is a problem for grain amaranth and seed shattering also. These are the breeding constraints these are the constraints in the moment that we try to address through breeding. For breeding, we need access to the biodiversity of the crop. The biodiversity of the crop we find generally in genebank collections. The World Vegetable Center holds the 4th largest Amaranth germplasm collection in the world. We have around 1,000 accessions of 18 different species. The germplasm is held in our genebank in Arusha in Tanzania, and we have also a copy here in Taiwan in our genebank at the headquarters. What our genebank is doing, it is collecting germplasm of important vegetables such as Amaranth. It is conserving this germplasm. It is characterizing the germplasm, and it is promoting the use of this germplasm either directly by farmers or by breeders. When we talk about use of germplasm, if we have a large germplasm collection, the use might be constrained because it might be difficult to screen 1,000 or more accessions for certain traits, especially if these traits are a little bit more complex. Therefore, we are proposing core collections as collections that represent the diversity of a larger collection in a much smaller germplasm set. The production of a core collection is illustrated on this side of the slide, where a large collection is characterized, and we are choosing a smaller portion of this collection that represents the diversity that is available here. How we made the core collections that I'm talking about in this presentation, I will tell you in about a minute. But before, please let me point out that we also have generated a magic population. This is a population that is derived from founder lines. We have identified 8 founder lines based on their yield, on their agronomical properties, on disease and pest resistances, nutritional properties, etc., etc. All traits that are important for breeders. These founder lines are crossed with each other, and the crossing products are again crossed with each other until the alleles of these founder lines are mixed in different lines. Then these lines are purified through single seed descent, and this results in a new allele combination, in allele combinations that are probably not available in the germplasm collection or in the nature. Like that, we can combine traits with each other in a very efficient manner. For example, good agronomical properties with disease resistances. This kind of population is also very useful for research and for mapping traits. But first, to our Amaranth core collection. Here I outline quickly the process, how this was done. We grew out our collection, we harvested a few leaves, and we extracted DNA from each accession. The accessions were then genotyped, and the genotypic data were analyzed. We were calculating an Euclidean distance matrix, and we were we're analyzing the diversity, and based on the diversity, we were grouping these accessions. And then we applied a method that was published by Odom et al. That uses the distance, the genetic distance between individual accessions to select those accessions, first of all, that group with each other, and then from each group, that accession that has the minimum average distance to the other accessions of the group. We can predetermine the number of groups that we want to have. And we have generated two core collections, one core collection for the whole collection. As I said, the whole collection had around 1,000 accessions. So we have chosen 10–15%, more exactly 12%, or 123 accessions to represent the whole collection. And for a second collection, where we focus just on 4 species that are of particular interest for Africa, we have decided to make a core collection consisting of 70 individual accessions. These core collections were established, but before, let me report you the results from the genotyping. After genotyping, we made a diversity analysis and we established a phylogenetic tree. The phylogenetic tree partially also separated the different Amaranthus species from each other. For example, this large cluster here is all Amaranthus tricolor. This small cluster here is Amaranthus preto. But there were a few clusters where different species were mixed with each other, like here hypochondriacus and cruentus, and there was also a small part of caudatus mixed in the hypochondriacus accessions. This might be due to wrong species annotation during collection, during regeneration, during characterization. Sometimes it's not easy to determine the species of Amaranths. And from this very diverse germplasm set, we have selected a diversity set for 4 species with 70 accessions and a diversity set of 123 accessions, representing the whole collection. We analyzed these core collections with population genetic indicators like Shannon Index and Nei's expected heterozygosity, and it showed, as expected, that the core collections do not differ strongly in these parameters from the whole collection, which indicates that the diversity is widely conserved. Then we looked into the phenotypes of the whole collection and the phenotypes of the core collection, and again, it showed that a large proportion, 97% in the case of the whole core collection, was maintained in the core collection compared to the whole collection. So the phenotypic variation was largely conserved in the core collection. So we find approximately the same variance in the core collection like we find in the large collection. That means that these core collections are very useful for isolating individual lines, for selecting lines, for crop improvement, or for direct use for farmers. We just encountered one problem that I have mentioned already. The problem is that sometimes we found wrong species annotation. Here, for example, on this phylogenetic tree of the core collection of the 4 species that are important for Africa, we find one clade which contains all the dubious accessions that we have in this core collection. But there are other clades, like here or here, where different species are mixed with each other. And that's not normal. Normally, the phylogenetic analysis should well separate the species. So we are applying means how to identify possible wrong annotations, and we look at them and we correct the species annotation. One means to make this in a very efficient manner is to do structure analysis. And here in the bottom, you see again a structure analysis of the 4 species core collection, where one cluster is specific for dubius. This is the yellow one in the middle. One is overwhelmingly consisting of hypochondriacus, and then we have 2 clusters with problems, and then there's anyway one cluster, a total outlier, which is probably not the caudatus like indicated here, but another Amaranth species that does not belong to the 4 other species. Based on this genetic analysis, structural analysis, we can pinpoint those accessions that are likely wrong annotated for correcting the species annotation. Once the core collection in place, we can work with them. The first thing we did was an agronomical analysis of the core collection. We were just drawing them out and measuring agronomical parameters. Just very briefly, a few results highlighted from this evaluation. The evaluation was done twice, once in the hot season and once in the cool season. It shows that in the hot season, the overall yield is larger than in the cool season. And it shows that in the cool season, the released varieties do better compared to the germplasm than during the hot season. That means that for the hot season, for this kind of planting that was applied, there is still some room for breeding higher yielding varieties. We were harvesting this trial not at once. We were harvesting 5 times. That means that the plants were successively defoliated. It shows that the first harvest had always the lowest yield, even in the hot season, like in the cold season. Then in the hot season, the yield increased from all harvests to reach again a minimum at the 5th harvest. In the cool season planting, the yield remained lower over all harvests. At the last harvest, there were a few genotypes that did not yield at all, whereas other genotypes had relatively good yield. So here there's also a way to improve the crop. I have only a very limited time available to tell you the story about the core collection, so I reach already the conclusions. What I wanted to tell you is that the World Vegetable Center is holding a genotyped Amaranth collection with more than 1,000 entries. These entries are available for the public. We are currently still working on the species identification in this collection. We have established diversity sets, the diversity set for the whole collection and the diversity set specifically for 4 species with great importance in Africa. We have developed a magic population for breeding and research. We have at the moment only about 150 magic population lines ready. By the end of 2021, we're expecting that the whole set of 400 magic population lines will be available for breeding and research. And I gave you a very brief introduction into the field evaluation of diversity sets for breeder desired traits. I was just talking about yield, of course. During the evaluation, we have also measured many other traits. The detailed results will be published very soon. At this point, I would like to acknowledge the people who did the work for this study. This was our team in Arusha, Tanzania, our team at the headquarters. The genotyping was done by Diversity Array Technology in Australia, led by Andrzej Kilian. The core collection was established by Richard Finkers from the Wageningen University, and the funding was obtained from the German Federal Ministry for Economic Cooperation and Development and from the Council of Agriculture, Taiwan. If you have questions, please use this email to ask, and if you want to know more about the World Vegetable Center, please visit our webpage, which is noted here. Now, I thank you very much, and I wish you an excellent day. Goodbye.